Hello, I'm Indira Carey, Marketing Manager at Origene, and today I'll discuss a new product we've launched called CytoSections. CytoSections are reproducible and renewable controls made for IHC, ICC, and ISH. Here's an overview for our discussion. We'll first look at challenges faced with current controls. Then we'll look at what CytoSections are and dive into why they're a unique solution. Finally, we'll look at how CytoSections can be used in your lab. Tissues, cell lines, and knockouts are used for positive and negative controls and for assay development. There are several challenges with each method. Finding tissue expressing or not expressing your target of interest can be difficult. In addition, there can be tissue variability lab to lab or even changing expression patterns when sectioning. Cell lines may show changes in expression over time, and knockouts may not be 100% nor permanent. Unlike tissue, CytoSections are a reproducible and renewable source. They save you on time spent searching for positive and negative tissue. Some tissues are valuable and difficult to obtain in enough quantities, so CytoSections allow you to save on the use of precious tissue for assay development. What then are cytosections? A plasmid expressing your cDNA target of interest is transiently transfected into mammalian cells. The cells are cultured for 48 to 72 hours and then harvested. They're processed as though they were normal tissue in a FFPE block and then sectioned. Sides are sent to you unbaked, so you're able to bake them according to your typical protocols. In fact, cytosections are readily incorporated into your usual workflows for IHC. Where do the cDNAs come from? Origene has an extensive library of cDNA clones. We have more than 20,000 human genes available in our library to make cytosections for your gene of interest. Origene's clone collection is well known and widely cited in the literature. Our clones use mammalian vectors that are optimized for robust protein expression, which ensures that your target is readily expressed and detected in the corresponding cytosection sample. Cytosections are generated using the same paraffin embedding process as tissues, which means cytosections are easily incorporated into your current workflow. During production, each lot is checked by Western blot and using a typical IHC workflow, so you're able to confidently use cytosections in your assay, knowing that expression is validated. Here's an example showing multiple target genes successfully expressed in cytosections. How does localization of the target in cytosections compare to tissue? On the left is BRG1 expressed in kidney tissue. It's localized to the nucleus. And on the right is an example of BRG1 expressed in cytosections, also showing nuclear localization. This is an example with colon tissue showing that the DCC protein is found in both the cytoplasm and membrane, which is also observed in the cytosection sample. Cytosections can be used for multiple applications instead of tissue samples. They're especially useful for antibody specificity studies and for controls. They can be used for discovery or antibody screens, as well as immunofluorescence and in situ screens. For more information, please contact our expert technical support team at techsupport@origene.com.